Hello students, today we are going to study about cholesterol and sphingolipid, its structure and biological importance in human body. Cholesterol is usually considered as a negative thing. People want to lower their cholesterol, but it's essential for human body. Essential for the proper functioning of cells' hormones. What is cholesterol? It is an important form of lipid that is required in our body. It is a soft, fat-like, waxy substance found in all the cells of the body. Then from where do we get cholesterol? There are two sources of cholesterol. Liver produces 75% of cholesterol and remaining you can get through your diet. Large amount of cholesterol is present in food like meat, fish, egg, butter, cheese, and milk. It is an essential structural component of cell membrane. In blood and plasma, cholesterol is transported by lipoproteins. Then come to structure of cholesterol. Structure of cholesterol contains two parts, one is steroid nucleus and another is hydrocarbon chain. Structure of steroid nucleus consists of 4 fused benzene rings. The ring in steroid are denoted by the letters A, B, C and D, with the carbon number in sequence. Steroid has O, hydroxyl, group at the third position of ring A. It is known as steroid alcohol or sterol. Though structure of steroid part contains alcohol functional group, so it is hydrophilic in nature. That means water-loving. The second part is long-branched hydrocarbon chain, which is attached to D-benzene ring in the diagram. And this long chain hydrocarbon are hydrophobic in nature. That means water-repelling in nature. Next one is types of cholesterol. There are 3 types of lipoproteins. These lipoproteins are a combination of fat and protein. Here lipids need to be attached to the proteins, so they can move through the blood. The 3 types of lipoproteins are: HDL stands for high-density lipoproteins, LDL stands for low-density lipoproteins, VLDL stands for very low-density lipoproteins. Now let's discuss these 3 different types cholesterol one by one. Here we will compare and understand what is high-density cholesterol and low-density cholesterol. High-density cholesterol is also known as good cholesterol, because it helps to remove excess cholesterol from your cell, tissue, and plaque from your blood vessel. High-density cholesterol returns back the excess cholesterol from blood to your liver, where it broken down and removed from your body. That means high-density cholesterol help to maintain your heart rate. Come to low-density cholesterol, which is also known as bad cholesterol. LDL carry cholesterol and travels through your bloodstream to the cell that need it. If your body has too much of low-density cholesterol, then it can build up in the wall of your artery. Low-density lipoprotein and other substances in the artery wall form a fatty deposit. You can see the yellow portion in the diagram. Over the time it can erode the artery and reduce blood flow. In this diagram, left side is the normal artery and right side is the narrowed artery. This is due to the bad cholesterol. It can cause coronary artery disease. Now we'll see the difference between low-density lipoprotein and very low-density lipoprotein. In case of very low-density lipoprotein, the plasma lipoprotein produced by the liver contains relatively large amounts of triglycerides compared to protein. VLDL composed of 10% of cholesterol, 70% of triglycerides, 10% of proteins and 10% of other fats. VLDL transports triglycerides from the liver to the adipose tissue. Whereas in case of low-density lipoprotein, plasma lipoprotein composed of a moderate proportion of protein with little triglyceride and a high proportion of cholesterol. LDL contain 26% of cholesterol, 10% of triglycerides, 25% of proteins, and 15% of other fats. LDL transports fat molecules around the body. VLDL is also known as bad cholesterol because it leaves a residue of cholesterol in the tissues during the process of conversion to LDL, which causing the narrow of artery. Next topic is about sphingolipid. It is the second largest class of phospholipid. The most distinct difference between sphingolipid and glycerophospholipid is that the backbone of sphingolipids is not glycerol, it is sphingosine. From the diagram you can easily able to understand the structure of sphingolipid. Sphingolipid contains sphingosine bases, which is a long chain of aliphatic amino alcohols, and a long unsaturated hydrocarbon tail, containing 18 carbon atoms, which contain a polar domain that includes amino group. These compounds play important roles in signal transduction and cell recognition. In the second diagram, R is the long chain hydrocarbon having the unsaturation. NH2, that is amino group, can attach to any fatty acid through the formation of ester linkage. OH group can attach polar head group, which are hydrophilic in nature. Now we will discuss the derivatives of sphingolipids, that is ceramides. These are family of lipid molecule, which are found in high concentrations within the cell membrane of eukaryotic cells. If you see the structure of ceramide in the diagram, the amino group of sphingosines can form an amide bond with fatty acid carboxyl, to give a ceramide. That means a ceramide is composed of sphingosine and a fatty acid. Sphingosine, which is a long chain amino alcohol with carbon-carbon double bond. Fatty acid is joined to sphingosine via an amide linkage rather than an ester linkage as seen in glycerol. Biological importance. Ceramide can participate in a variety of cellular signaling. It is a component of cerebrosides and gangliosides, which are essential for the proper functioning of brain cells. It play important role in coordinating cellular response to extracellular stimuli and stress. It prevent excessive water loss due to evaporation as well as barrier against the entry of microorganism. Thank you, and for further clarification and doubt please let me know.